Hey there, and welcome to this explainer. So, if you're a biology student, or honestly just someone who's fascinated by how life actually works, you've probably stared at one of those confusing textbook diagrams of cellular energy and felt, well, completely lost. Trust me, we've all been there. But today, we're throwing the textbook right out the window. Okay, let's dive into this by transforming those complex, invisible cellular processes into a relatable, moving, working tour of your very own household kitchen.
Look, it is completely normal to feel totally overwhelmed when you're trying to memorize the massive, multi-step machinery of cellular respiration. All those molecules, the endless cycles, the protein complexes, it can literally look like alphabet soup. But what if I told you that your cells operate exactly like a highly efficient, fast-paced culinary team prepping a massive feast? By the end of this explainer, you're not just going to memorize this process, you're actually going to see it functioning right in your mind's eye.
All right, before we start cooking, we need to talk about money, specifically the biological currency that pays for all the physical chores around your cellular house. This currency is called ATP, or adenosine triphosphate. You see, every single piece of food you eat, whether it's carbs, fats, proteins, you name it, it all must eventually be broken down and converted into this exact molecular currency to get any actual physical work done.
Now check out the structure of ATP itself. Notice those phosphate groups all lined up. The bonds holding those phosphates together, those are classified as high-energy bonds. Think of them like tightly coiled, loaded springs, or maybe a stash of stored cash just waiting to be spent. When the cell needs energy, it simply snaps off that end phosphate, releasing a huge burst of free energy to power whatever chore needs doing.
Section 1. Prepping groceries with glycolysis.
Let's move right into our first major step. We are starting with the messy prep work, bringing the groceries into the house and unpacking them on the kitchen counter, which in our cell is the cytoplasm. This gives us a beautifully clear macro view of our entire cellular floor plan. Picture glucose, our main grocery item, entering the cytoplasm. This is where the initial breakdown happens. And it's absolutely crucial to realize that this prep work takes place entirely outside the mitochondria, which is our main cooking stove that we'll visit in just a bit. The ingredients have to be processed on the counter before they ever get near the high heat. Let's zoom in a bit and watch this prep work actually happen. We've got a 6-carbon glucose molecule entering the preparation phase. Through a super rapid sequence of enzymes, just like a master chef rapidly dicing up raw ingredients, the cell actually spends a little bit of ATP to chop that glucose right down the middle. Then, moving into the payoff phase, it splits into 2 3-carbon pieces. We call these pieces pyruvate. So, let's check our running energy ledger. By the end of this cytoplasmic prep work on the counter, the cell has netted exactly 2 ATP. Now, sure, it's a small paycheck, but it's the critical first step in our metabolic cooking process.
Section 2. Mitochondria, the kitchen stove.
Okay, now that our ingredients are perfectly prepped into those little pyruvate slices, they need to be transported into the specialized high-heat cooking zone. When we compare a real electron micrograph to a mapped diagram of the mitochondria, you can really see how complex the inside is. It's definitely not just an empty sac. It has an outer membrane, an inner membrane folded into structures called cristae, and an inner fluid called the matrix. Think of these as highly specialized kitchen zones, keeping our culinary magic totally organized and efficient.
But hey, don't think of these stoves as static chunks of metal. They are incredibly dynamic. Over a span of just 20 minutes, these cellular kitchens actively remodel themselves. They can divide into smaller pieces, a process called fission, or they can merge together into massive interconnected networks through fusion. They're actively adapting their size and shape based entirely on the cell's immediate energy demand.
As we transport our prepped pyruvate from the cytosol into the mitochondria, watch this transition. A transport protein actively pulls it inside, and right as it enters the stove, boom, it's oxidized. It binds to coenzyme A to form acetyl-CoA. During the specific chemical chop, we generate our very first piece of biological kitchen trash. Yep, a molecule of carbon dioxide is released and exhaled away.
Section 3. Cooking energy, the Krebs cycle. With our acetyl-CoA primed and ready, we enter the actual simmering phase of our cooking process. Imagine this cycle continuously running in a loop within the mitochondrial matrix. Our acetyl-CoA drops into the pot, reacting with a resident base ingredient called oxaloacetate to form citrate. As this molecular stew slowly simmers, high-energy electrons are carefully extracted. These electrons get stuffed into specialized carrier molecules called NADH and FADH2. Oh, and notice those bubbles popping off? That's more carbon dioxide waste being tossed right into the kitchen trash.
Time to update our running ledger. For every molecule of original glucose that goes through this simmering Krebs cycle, the cell earns another net gain of 2 ATP to spend on its household chores. But honestly, the real treasure we've collected here isn't the ATP. It's actually all those loaded electron carriers we just filled up to the brim.
Section 4. The Conveyor Belt Electron Transport Chain.
And this brings us to the final, fastest, and frankly most amazing stage, oxidative phosphorylation. I want you to imagine this as an incredibly rapid, highly pressurized food delivery conveyor belt. We have 4 massive protein complexes embedded right into the inner mitochondrial membrane. Watch the action here. Those loaded carriers we talked about, NADH and FADH2, speed up to the belt and drop off their high-energy electrons. As these electrons bounce rapidly down the complexes, their energy is used to actively pump protons, those H+ ions, straight up across the membrane. This creates a massive, highly pressurized reservoir of protons trapped in the inner membrane space.
And this brilliant mechanism, originally discovered by Peter Mitchell, leads to the ultimate payoff. Enter ATP synthase. Because we built up that massive proton pressure, those protons literally rush back down through the ATP synthase structure. This causes a physical rotation of the protein. It functions exactly like a turbine spinning in a dam, just churning out massive amounts of ATP as it turns.
Let's lock in the rapid-fire sequence of this conveyor belt. 1. Electrons rapidly move down the chain. 2. Their energy pumps protons to create a high-pressure reservoir. 3. We need a final ingredient to catch those falling electrons at the end of the belt, and that ingredient is oxygen. 4. When oxygen catches those electrons and protons, it gracefully turns into completely harmless water. It's a perfect system.
Section 5. Final yield in power outages.
Let's bring this entire metabolic journey together. But first, we really need to ask a critical question. What happens to this busy kitchen if it unexpectedly loses power? Here is the stark contrast. When the cell has plenty of oxygen, we call it aerobic conditions. The kitchen is fully operational, the stove is hot, and that conveyor belt runs perfectly. But if there's a power outage, meaning no oxygen, we're in anaerobic conditions. The main stove literally shuts down because there's nothing to catch the electrons at the end of the chain. So the cell has to fall back on an emergency backup plan right there on the kitchen counter, directly converting pyruvate into lactate just to keep a tiny trickle of energy flowing.
But hey, when everything is working perfectly under aerobic conditions, we get this master chemical equation. Just look at how beautifully it balances out. 1 molecule of glucose plus 6 molecules of oxygen undergo a massive oxidation and reduction process. They perfectly transform into 6 molecules of carbon dioxide trash, 6 molecules of water, and pure, usable energy.
So, what is the final tally of that energy? Well, because of that incredible spinning turbine of the electron transport chain, one single humble molecule of glucose yields a grand total of 36 molecules of ATP. I mean, that is a massive return on investment from our original grocery delivery, right?
You've followed the ingredients from the prep counter into the high-heat stove and all the way through the turn-in conveyor belt. You now actually know the intricate mechanics of your own life source. So I'm going to leave you with this final thought. With trillions of these tiny kitchens working at lightning speed inside you right now, generating millions of ATP every single second, what complex chores will your cellular kitchens empower you to accomplish today? Thanks so much for joining me on this explainer, and keep exploring.